So, hi, good morning. Good morning, everyone. So, am I audible? My voice is reaching to you? Good morning, good morning. Good morning. I'm all good. What about you? Because obviously for you it's really good because you are at your own place enjoying with the friends. At the same time, you are doing your task also, like you are studying also. So somehow you are ahead of your friends because other friends are doing what? Only enjoyment, but you are doing enjoyment as well as study both. And just keep it secret. Reveal once this month, or we can say 2 months is done. So reveal it like I was studying as well as enjoying with you also. Okay, good morning, good morning. Good morning, Varsika, Dheeraj, Himansika, Maheshwari. Good morning, Namaste, Vijay, Srija, good morning. Good morning, good morning. So I think Vanakkam, yes, I'm all fine. Vanakkam. So anyway, in the last class, we have discussed something about what? Prokaryotes. And even we have started with the prokaryotes. What is prokaryotes? So pro means primitive, outdated one, or we can say the older one. And karyote means what? Karyote means it's a nucleus. So the older nucleus, outdated nucleus, primitive nucleus, that is known as prokaryotes. So anyone who is having the prokaryotic cells is considered to be the prokaryotic organism. Okay. And mostly the prokaryotes are what? Good morning. Krishna, shape of bacteria also done, it can be of four types. One is spherical, other one is what? Rod shape. Other one is comma shaped, and next one is a spiral one. So we can have a lot more. And other things we also know that cyanobacteria, blue-green algae. So these are the few names we are giving to this. And why we should be thankful to the cyanobacteria? I think we have discussed in the last class. Why always and always we should be thankful to cyanobacteria. Because whatever let's say in my body, whatever nitrogen is there, so this nitrogen is coming from where? Nitrogen is coming from the cyanobacteria itself. No doubt, directly we are not taking nitrogen from the cyanobacteria. In some part it's there, but not always we are dependent on cyanobacteria for nitrogen. We are dependent directly on what? Plants. And plant depends on what? Cyanobacteria. And cyanobacteria is having magical power, miracle one, and that can fix the atmospheric nitrogen, convert into the nitrogen related compounds. Nitrogen related compounds, and that compound which we are eating, or we can say we are utilizing in day-to-day life. Okay, so in this class, just we'll talk about the structures of prokaryotes. And obviously, how the prokaryotes looks like. How prokaryotes looks like. Simple or complex? Simple or complex? How it looks like? So, I hope it's a simple one. But when it comes to the behavior, it's like a highly notorious, or we can say it's a highly complex one. It's a highly complex one. So we can say it looks simple, but don't go for the simplicity. It will be complicated for you when it comes to the behavior. So that's why be aware of what? Prokaryotes. Don't go for the looks, just go for the behavior. Because when you stay for a while, it will tell you how differently it can behave. How differently it can behave. So now we'll go to the structure of what? Structure of prokaryotes. And generally we are talking prokaryotes means it's a bacteria. So here our topic today's topic is structure of prokaryotes, or we can say it's nothing the prokaryotic cells. Okay, how to know the structure, or we can say it is microscopic in nature. So I am not able to visualize with my naked eyes. So how to visualize? How to study the structure? Because the structure which is not visible to me, how we can study? How we can study? So one scientist was there. His name was Graham. What is his name? His name was Graham, and he started studying the microscopic structure. Because obviously it's more than difficult to understand the structure of microscopic organism. But if you stain it, if you make it colorful, you can visualize it. So the first part that is coming to, it's nothing but the gram staining. So one scientist was there. One scientist, pretty good, and he gave a lot more contribution. What? He gave a lot more contribution to biology. So that's why lot more contribution to the biology, and his name was what? And Graham was excited with his work. So his name is what? His name is Graham. So one day when the Graham was going to his home, he found some plates, or we can say some utensils was there. And at the same time, he was also carrying the microscope. So he put the plate under the microscope. After that, he was seeing some small organisms are crawling. No doubt it was crawling, but it is not visible to him. Later on, he got an idea. Once we make it colorful, I can properly visualize it. I can properly study it. So he started using. So here we can say a lot more bacteria was there. So here it's a group of bacteria. Let's say it's a group of bacteria, and this group of bacteria is not visible to you. So this group of bacteria is not visible to you, but still you want to study. So what to do? Like Graham Stein, or we can say Graham was the scientist who started playing holy with what? Or we can say it's a colorful holy with what? Colorful holy with the microscopic organisms, or we can say it's a bacteria. After that, what will happen? So here we can say he just used the alkaline solution of what? Alkaline solution of that we can call it as a crystal violet. It's a kind of chemical substance. So this chemical substance, you have to dissolve it into the solution so that you can pour this crystal violet solution over this easily. Okay. After that, we'll move to the next part. After doing this, sometime what's happening, more bacteria becomes more colorful. If it absorbs more color, then again it will be problematic for you to understand, or we can say to study the structure. So let's say this is the slide, or we can say it's a glass slide. Over that glass slide, it's a bacteria. Over that, it's a bacteria, and I just poured the color over that. But again, what will happen? This is the microscopic. This is the microscopic organism. CSK7, we have already done with the prokaryotic cells and eukaryotic cells difference. Okay. So again, we'll go to this topic. Here it's a prokaryotic cells. Later we will start with the eukaryotic cells. So it's a microscopic organism. And this is the glass slide. Over the glass slide, I just place the microscopic and just put it over the flame. Put it over the flame so that this will make a smear, or we can say it's a layers. This makes the layers. After that, what I can do is I just started pouring what? Crystal violet color. Crystal violet color. So this crystal violet color, sometime what happened, like too much of color poured over this. Now it is not visible to you. So after that, what to do? Just take we can say drop of water and place over that, so that excess of color will go away. Only the small part of color that is attached to its body, will remain with it. So what to do next? Tell me. Wash with the what type of water? What type of water? So it's like a crystal clear water, or it's a polluted water. So obviously it should be distilled water, or we can say it's a crystal clear water. Okay. So here we can say wash with what? That is distilled water. After that, what will happen? So after this crystal color, so it becomes what? It becomes purple, or we can say it's a violet in color. Okay, crystal violet, or we can say blue in color. Thereafter, what to do? Again, we have to put some kind of iodine, or we can say it's a potassium iodide. We will treat with it. That is potassium iodide. Now we'll treat with the potassium iodide, and this potassium iodide, it is acting as what? It is acting as a moderant. So what is the uses of this Ki, or we can say potassium iodide? So let's say I bought one cloth from the market. But after, let's say this is the color. But after 2 to 3 wash, so this becomes white in color, or we can say totally white in color. Why? Because the company, or we can say from where I got this clothes, they have not used this mordant properly. And if they are not using this mordant properly, what will happen? So this cloth will not absorb the color completely, or we can say properly. So that's why we should use this mordant properly with the clothes. Otherwise, your clothes will fade after some time. Okay. Now, next one is, after that, what to do? After doing all those things, we have to wash with either, like if you are having the acetone, it's fine. But if you don't have acetone, then what can you use? So you can use the absolute ethanol. You can use the absolute ethanol, or we can say it's nothing but the ethanol. So after doing this, what will happen? Few bacteria remains colorless, and other bacteria absorb the color, or we can say it becomes colorful. It becomes colorful, and obviously the color would be, we can say blue or violet color. Now just tell me, which one is gram negative and which one is gram positive? Firstly, the colorful is gram positive because it is behaving according to the gram, and if it is not behaving according to the gram, it is negative. So one is positive, and one is negative. Who is negative? The one who loses its color. The one who loses its color, that is known as negative, and the one who retains its color is known as positive. So is that clear to everyone? Who is positive and who is negative? Who is positive and who is negative? So the white one is negative or positive? So obviously it's negative, and the colorful it's a positive because it is behaving according to what? According to the gram. So whoever is following, or we can say obliges, or we can say it's obedient to gram, that is considered to be as a positive. If you are not following the gram, you are considered to be as a negative. So one is negative and one is positive. But still, who is sad? Gram negative or gram positive? Who is sad? So I think gram negative is sad. Why? Because gram negative loses its color. So after losing the color, what will happen? There is a sadness. You can see the sadness on its face. Okay, but still, gram was too much, we can say what, optimistic. So he said that don't worry, I'm just giving you some other color where you can become colorful. You can become colorful. So that part is, so firstly we'll write one thing. It is considered to be as a gram negative, and other one is considered to be as a gram positive. So gram negative means what? The bacteria lost its color, or we can say not able to retain the color. Here it's nothing but the retain the color, or we can say absorb the color. Retain the color, or in other word we can say that is one is colorful and one is colorless. But again the gram is having some different idea for whom? For the gram negative, because gram can only study the structure of that bacteria who is colorful. Gram is not able to study the structure who is colorless. So obviously it can go for coloring the colorless, or we can say it's a gram negative one. So let's move to next. Gram needs to color it, but crystal violet is not more than enough to color. So they have used some other color, or we can say other dye. The name of the dye is nothing but the safranin. The name of the dye is nothing but the safranin. After getting this dye, how the gram negative looks like? So the gram negative looks like either it's a pink in color, or we can say it's a red in color. So red or pink in color it looks. Now the gram is also happy. The bacteria is also happy, like bacteria becomes colorful, and gram was happy because gram can properly visualize it, study it, and publish its a paper so that it becomes popular in the area of microbiology. Microbiology. So here it becomes colorful. It becomes colorful, or we can say it's a pink in color, or we can say it's a somehow orange in color. Okay. So here it's I will do that GLK, like Lochan GLK. I will do this at the end of the class. Okay, so it's like we can say it's not. Don't confuse with this red or pink. Like sometime it is having the more, or we can say intensity of color absorption is more, so it becomes red. If it is little bit less, then we can say it's a pink. That depends. That depends. After that, what to say? Like the gram was excited. Who was excited? Tell me, who was excited? So here I can say that so gram was excited to study what? Study the structure of microscopic organism. Okay. So Gram was excited with this experiment, and he started doing what? He started doing the study of the whole bacteria. Now firstly I will draw the diagram of bacteria, and in this case earlier it was gram negative, means it's a colorless. Now it becomes colorful, and in this case the gram positive, already it's colorful. So gram was doing good with the bacteria both, that is a gram negative as well as gram positive. So directly we'll go to the next what? So is there any problem with this? Here it's like a Swarup Reddy, why gram negative bacteria absorb safranin but not crystal violet? See, because the color, or we can say the composition, chemical composition of each and every organism is different. Each and every organism is different. So you like some other color, other one looks, or we can say other one likes some other color. So the absorption, or we can say capability of absorbing the dye, depends on what type of chemical present inside your body. If the chemical inside your body can interact with the safranin, you can absorb safranin. If the chemical is not there, you are not able to interact with that chemical, but it doesn't mean that you are not interacting with any other color. You can interact. You can interact. So is that clear? Like why not all bacteria is absorbing same type of color? Again, same thing I have repeated, like I have explained, Lakshmi Duveri, like why the safranin is not used in gram positive, because the chemical composition is totally different. Chemical composition is totally different. So that's why you can't use this. You can use the safranin, but at the same time absorption is very less. Absorption is very less. Because let's say the choice of the clothing colors is different for different person, or we can say similar for all the person. Because some person wants the light in color clothes, other one it's a bright in color. That depends on you. That depends on you. Okay. Is elite and elite star batches different? Obviously, in both the batches, same teachers are going to teach. So actually, one more, Hasni, ready? Why he used only saffronin? It's not like he used only saffronin. He has used a lot more colors. Because whenever we are studying about the scientist, we are not studying about the failure, or we can say we are not studying about the what are the experiment he has attempted. So only the part which we are studying is the one in which he got the success. He got the success. Okay. So because after becoming doctor, is there any chance that your patient will say, or we can say, will ask the question, how many times you have attempted the NEET exam? Or we can say, can a doctor will make a poster. In that poster, he said that, or we can say the doctor will write like this: I have attempted 5 times in the NEET exam, and at the 6th attempt, I got the seats. Obviously, no one will do that. No one will do that. So everyone is writing about its success, not the failure, right? So shall we move to the next? Now we'll go to the structure of what? Structure of prokaryotes. Chalo, theek hai. Let's say this is the bacteria. Is that visible? The bacteria which is in my hand. Is that visible? Okay, good. Means you are having some kind of miracle type of personality that you can visualize the bacteria, because bacteria is microscopic in nature. I just zoom in, or we can say make it colorful so that it is visible to you. Is it? It's visible to you. Now I will do one thing with the bacteria. I will start studying the bacteria. If I am studying, or we can say you are studying the bacteria, the outermost layer in the bacteria, that we can call it as a glycocalyx. So here I am writing one, or we can say I am drawing one bacteria, and in this bacteria, the outermost part is what? Outermost layer of bacteria. Outermost layer of bacteria is what? It is known as glycocalyx. And in this glycocalyx, what will happen? So glycocalyx will somehow give protections in different manner. That's a great Usani, glycocalyx. So what's that? It's a glycocalyx. So in the first part, and just below the glycocalyx. So here what I have said, the outermost layer is known as glycocalyx. After that, one more question, human. Is that your ID? Human, no doubt you are human beings, but still, how can he separate two color and colorless two color and obviously, why not? You can separate. Can you separate here the white color from this sky blue color? How to separate? Obviously, it's a physical quantity. So you can separate, right? So anyway, if you are moving, like I have seen this bacteria. In this bacteria, the first layer which I am finding is what? It's a glycocalyx. Now, just let's say inside the bacteria, and inside the bacteria, what are the layers he find? He said that no, it is considered to be the cell wall. It is nothing but the cell wall. Just inside that, he found another layer. The name of the layer is known as it's a cell wall. So the second layer he found, no doubt it's a microscopic, but we are having the, or we can say we are superpower. So we can visualize. So the second one is cell wall. Second one is cell wall. So how many of you are having the superpower? Just whoever is having the superpower, just thumbs up. I hope everyone is having the superpower, right? Because if you don't have superpower, how can you say that? Means how it's visible to you. Obviously, it's visible to you because you are having super power. And one more question. Oh, a lot more. It's like a kind of superpower class is there. So everyone is having superpower. Okay. Human. Now let's start with the other part. Like this is the cell wall. And we can't say that cell wall is only present in the plants. So plant is also having the cell wall. Fungus is also having the cell wall. Next one is the bacteria. So bacteria, plants, fungus. And according to you, how many kingdoms are there? How many kingdoms are there? Can you count the fingers? So obviously it's a 5. Out of 5, how many of them is having the cell wall? 1, we can say it's a Monera. Some of the Protista is also having the cell wall. Some of the Protista is also having the cell wall. Next one is a fungus is also having the cell wall. Next one is plant is also having the cell wall, but only one, that is nothing but the Animalia is not having the cell wall. Okay, is that clear? So here we'll go to the just inside the cell wall, which layer will come? So can you tell me that it's nothing but the cell membrane? Cell membrane. And we have already studied that cell membrane is the only structure which is present in all type of cells. If it is a living, then it is 100% present. If it is a living, it is 100% present. That is a cell membrane. Just keep in mind, don't forget this part. So after the cell wall, we'll move to the cell membrane. That is a quite interesting structure. It is selectively permeable and it can perform a lot more activities. So again, we should be thankful to the cell membrane. So I'm just drawing the cell membrane. So this is the cell membrane, but at the same time, cell membrane is having some kind of infoldings. Okay. So after that, again it will go like this. Again you can find a small infoldings or this type of structure. At the same time, there is a enlargement. Or we can say it's an outgrowth. It's an outgrowth of the cell membrane. Next one is, sometimes you can find a lot more outgrowth. So this is the second outgrowth. Next one is, if you just keep on adding here, you can find one more outgrowth. So a lot more outgrowths, or we can say a lot more structures that is related with the, or we can say that is the modification of the cell membranous structure, or we can say it's a membranous structure. Okay. So here we'll go to the next part. I will just level this whole diagram. So my first leveling is this. this whole infolding, this complete infolding, is known as, mesosomes. Okay. Acha, how many of you are having the mitochondria? I think your answer is everyone is having the mitochondria in your group, right? So everyone is having the mitochondria. But do this bacteria having mitochondria? Do this bacteria having mitochondria? Obviously, my answer is no. My answer is no. There is no such kind of mitochondria present in this bacteria. And what is the main function of mitochondria? Is to give what? Is to give huge amount of energy, tremendous amount of energy, or we can say it's a plenty of energy. That's why we can call it as a powerhouse. But this one is lack of powerhouse. This one is lack of powerhouse. But how it will survive? How it is surviving? How it is surviving? How it is surviving? It can survive with the help of this miracle thing, or we can say some magical structure. This magical structure is known as what? Mesosomes. Means mesosomes will do the similar things as it is done by the bacteria in my body, in your body, in the plant cells, in the animal cells, in the fungus. So can we say that mesosome is somehow equivalent to mitochondria of higher plants and higher animals, or we can say it's a higher organisms? So higher organisms is having mitochondria, but lower one is also having the similar structure that is known as mesosome. So is that clear? Is that clear? What is the function of, or we can say, what is the structure of mesosome? Why mitochondria is not present in the bacteria, but instead of mitochondria, what is there? It's a mesosome. Okay. Now we'll move to the next structure. If this mitochondria, if this bacteria, if this bacteria is what? If this bacteria is photosynthetic in nature. Photosynthetic means what? It can produce its own food and started eating. Or we can say, let others feed. Okay. Obviously, it will feed others as well as feeding by themselves. So whoever is cooking their own food, it should have some kind of a structure, and that some kind of a structure should be capable, or we can say is capable to trap the sunlight, convert into the chemical energy. So chloroplast is not present here. Likewise, mitochondria is not present here. Chloroplast is also not present here. So instead of chloroplast, what is there? One more part is there. That one more part is known as, what is that? Chromato. So this structure is known as chromatophores. Chroma means what? It's a color. Chroma means it's a color. So this colorful structure will catch the light, will fix the light, will absorb the light and convert into what? It's a chemical energy. And this chemical energy will help in cooking the food. So is that clear? After that means two structures that is somehow related to the cell membrane. Next one is, one more structures will come, and this structure is known as okay, it's a buffering. Or is that clear? Like my voice is reaching clear? It's clear? Okay, good. Don't worry, it will. So here, what is the function of pili? So pili mainly participate in what? Pili mainly participate in reproduction. Reproduction, or here one method they are following. That method is known as what? That method is known as conjugation. That method is known as conjugation. So one bacteria comes in contact with the other bacteria, they will exchange the genetic material. They will exchange the genetic material. And after that, they become, we can say, rejuvenate, or we can say they will start reproducing. So if the question is coming, how this pili helps? Can we say that pili helps in reproduction? Or we can say pili helps in conjugation. So this is nothing but the pili which helps in conjugation. Okay. Or we can say it's a reproduction, or we can say exchange of genetic material. So how many of you have seen the male bacteria and female bacteria? Male bacteria and female bacteria? Male bacteria and actually, one thing is it is not visible, but if it is visible, then based on one features, we can say that this is male bacteria and this is the female bacteria. And whenever the male bacteria comes in contact with the female bacteria becomes male bacteria. But obviously this property, or we can say this is not applicable to all bacteria. But if it is there, it can, like a male bacteria when comes in the contact with the female bacteria can become the male bacteria. Okay. So anyway, firstly we'll come here. Don't worry, pili part, or we can say rest of the part we will explain. Just I am asking one question. So what is the function of pili? It helps in reproduction, or we can say it helps in conjugation. It helps in conjugation. Okay. This part of bacteria, what's that part? This one. This pili will help in what? Pili will help in conjugation. Pili will help in reproduction. No doubt a lot more pili is there, and pili is nothing but the outgrowth of the cell membrane. Outgrowth of the cell membrane, and it helps in exchanging the genetic material. Okay. Yes, I am getting the correct answer, that is a conjugation. Conjugation means that is exchange of genetic material between the two bacteria, that is known as conjugate. In conjugation, physical contact is required. Physical contact is required. Now we'll move to the next one. So next one is known as fimbriae. So what's that? It's a fimbriae. Let's say this is a bacteria. This is a bacteria. Now, how this bacteria will attach to my body? How this bacteria will attach to my body? With the help of this structure that is known as fimbriae. So fimbriae will help to attach to the, let's say it's a board, my skin, or we can say the soil, or any other parts. So it can attach to any other surface, or we can say any surface, with the help of this what? With the help of this fimbriae. So is that clear? What is the function of fimbriae? What is the function of fimbriae? So fimbriae, it helps in attachment to the host, or we can say attachment to any surface. Attachment to the any surface. Okay. Now we'll move to the next one is, let's say, so this is the part. So this whole part we can call it as a flagella. This whole part is known as flagella. If bacteria wants to go from one place to another place, let's say the bacteria is moving from where? Moving from Bangalore to Chennai. Bangalore to Chennai. What will happen? So what will help? So here we can say flagella will help the bacteria to move from one place to another place, and anyway this helps in motility, or we can say it helps in locomotion. It helps in locomotion. Okay, so is that clear? Now we'll move to the next. So what's the next? Now it will enter inside the matrix. Will enter inside the matrix. So what's there in the matrix? The most important thing is it is having double stranded but circular or linear. So here we can say it's a double stranded circular DNA. Double stranded circular DNA, and DNA is considered to be as a genetic material. So what's a DNA? DNA is considered to be as a genetic material, and there is no end, so we can say it's a circular. But in my body, whenever I am seeing this seeing this cells, and once you rupture these cells, you can find your DNA as what? DNA is having two ends. So in my body, what type of DNA is there? That is the linear DNA. But in the body of this bacteria, what type of DNA is there? So that is nothing but double stranded, means one strand. This is the second strand, but it is circular in nature. So here we can say double stranded circular DNA is present. Okay, so is that clear? Now the next part is, here we can say double stranded but circular or linear. So here it's a double stranded circular DNA is present. Double stranded circular DNA is present. After that, what to do? After that, I find one more structure that is unique. Like how one bacteria is looking from other bacteria. Or we can say, what are the structures which gives unique identity? So that structure is known as what? It's a plasmid. Great. Hasani Nagireddy, plasmid. Yeah, your question itself answering. So this is nothing but the plasmid. So another structures which I can draw here is its name is plasmid. Its name is plasmid. So can we say that one bacteria is looking from other bacteria? One bacteria is looking different from other bacteria, or the structure which gives uniqueness to the bacteria. This structure is known as what? This structure is known as plasmid. And plasmid is having what? DNA or RNA? So my answer is it's a DNA. Okay, is that clear? Two structure which is having DNA. One is it's a bacterial genome, or we can say this is the double stranded circular DNA, and this is also DNA that is known as plasmid. So can we say that plasmid is extra chromosomal DNA? This is the chromosome which is having DNA, but one more structure is there that is also having DNA. So can we say that this structure is extra structure? This structure is extra structure, and this extra structure is also having DNA. So now tell me, plasmid is known as extra chromosomal DNA. True or false? True or false? So I think my answer is true. And this gives you unique phenotype. By having different type of plasmid, you can see yourself differently. Okay. So is that clear? So these are the two structures that is most important here. After that, one more structure is there. So that will help in synthesis of what? Protein. Synthesis of protein. So can we say that ribosome is the structure which helps in synthesis of protein? Okay, so here it's a one more structure will come, and that another structure is nothing but the ribosomes. And ribosome is having one is chinna and one is pedda, or we can say one is bigger and one is smaller subunits. So two subunits are there. One is bigger and one is smaller subunits. And this mix up what? So this mix ribosomes, and combinedly the whole ribosomes, we can call it as a ribosome, but it's a what type of ribosome? That is 70S type of ribosomes are there. 70S type of ribosomes are there. Okay. Now after that, we'll move to the next part. One more answer is what is single, or what is difference between single and double stranded DNA? Strand means what? Strand means like this is the strand. This is the strand. If I am having one more strand, it becomes double stranded. So sometime it's only one string is there, and other time it is having two strings which are folded upon each other to give your double stranded part. So one is single stranded, one is double stranded. So you are having single stranded or double stranded DNA? Dinesh, you are having single or double stranded DNA? You are having single stranded or double stranded DNA? Obviously, you are having the double stranded DNA, and that double stranded DNA is circular or linear? Circular or linear? That is present inside your nucleus. Inside the nucleus, it's a double stranded linear DNA is present, but in your mitochondria, again it's a double stranded circular DNA is present. In my mitochondria, what type of DNA is present? In my nucleus, what type of DNA is present? Double stranded, but it's a linear. Okay. So, is that clear up to this part? Now we'll move to the next one. So, what is the difference between the circular DNA? Not much difference. In this case, double plasmid, again it's a double stranded DNA is present. But plasmid is giving uniqueness. Plasmid is giving uniqueness. After that, the bacteria is also having some kind of bacteria. What does it mean? 70S. So here it's a 70S. S is nothing but the Schwarzberg unit, sedimentation coefficient, Schwarzberg unit. Navya, Nikita, I think same question is coming. 70 is the number. 70 is the number. And the next one is S stands for what? S stands for the sedimentation coefficient. Sedimentation coefficient, or we can say it's a Schwarzberg unit. This will give you the density of ribosomes. Like I am measuring the distance. You are measuring the distance in which unit? Kilometer, meter, or something like this. Same as it is, if you are measuring the density of this ribosomes, you can measure in what? We can measure in what? That is a sedimentation coefficient. Sedimentation coefficient. So that's why I'm writing 70S. Means this one is having this type of density. Some ribosome is having 80S type of ribosome. Means 80 density is there. Okay. So is that clear? Is that clear? What is sedimentation coefficient? Sedimentation means what? It is settling down, settling down, settling down, settling down. So whenever you are pouring what? Sand or stone in the water, what will happen? The sand or stone slowly settle down. So this is sedimentation. This is nothing but the sedimentation coefficient. So this coefficient will give you the unit. Okay. Good. Sedimentation coefficient, Sandhya. It is same as sedimentation coefficient which you have written. Okay. So is that clear? Hasini, like density of ribosome in my body, it's 80s. But in the mitochondria, again, it's 70s is there. So 80s, 70s, S is nothing but the sedimentation coefficient. Is that clear? Okay. So now we'll move to the next part. So next part is it is also having some kind of a stored food, and this whole stored food we can consider it as a what? We can consider it as a inclusion body. We can consider it as a inclusion body. Okay. After that, what else we can write? So here I can draw one more diagram. In this diagram, I will firstly draw a string-like structure. This string-like structure is what? This string-like structure is known as mRNA. This string-like structure is known as mRNA. And this mRNA is known as messenger RNA. This is known as messenger RNA. Okay. Here it's an inclusion body. Means these all terms we will discuss in the next class. Today I have just drawn a picture. Thereafter we'll move to the. So here it's another word. So this is known as mRNA, and over this mRNA, what is there? Over this mRNA, a lot more ribosomes are present. So this whole structure is known as what? Polyribosome. So what is this polyribosomes? So polyribosomes is nothing but the green part is known as mRNA, and over that this yellow part is known as what? This one is ribosomes. And inclusion body is what? So inclusion body means what? Any type of chemical substance, either it's a stored food, excretory materials, or anything which is present inside the cytoplasm. This is known as what? This is known as inclusion body. Navya means this is the answer. What is this inclusion body? Any type of chemical substance, or we can say inorganic substance, present in this cytoplasm is considered to be as what? That is the inclusion body. Okay. So here is inclusion body. So till this point I will cover. Thereafter rest of the topic we will discuss in the next class. Or we can say we will go a lot more in descriptive way, that is all terms, in the next class. So till now we'll keep it, or we can say stop here, pause it. Pillai, or we can say, no, we can't say it's a pillars. It's a pillai is more than enough. What is mRNA? Lakshmi, mRNA is, M is a messenger. M is messenger, and this is the RNA, messenger RNA. Okay. Okay, so bye bye everyone. We'll meet in the next class. Hope this class is beneficial to you.
Hello?